Hi all, this is my second video on worm farming in Oman, and I wanted to give an update on my worm boxes, especially on the boxes that have baby worms that are grown from cocoons. So you can see how they advance and how they are different stages of growth that they have. Okay, so I will show you first in the same order of the boxes that I have. This is the first box with the worms I gather from the patio. These are worms that withstand the summer. They're quite big worms and they are soil worms, so I don't know exactly how well they will do in composting. So far, they seem to eat very quickly the what I add. Um, they do not try to escape. This is the watermelon that I added last week. There's almost nothing left, and yeah, so they're doing really well. They're really healthy. You can see that I think there's more than one type of worms here. I will do the classification later. A little bit more rest of watermelon. I don't see many cocoons though, so I don't know if they're going to reproduce. This is an interrogation mark here in this box. If these worms are going to be able to reproduce in this composting environment or not. Um, but so far they are doing really well and they seem to like all the food scraps that I add, and they are very healthy and big. Yeah, so this is a corn that I added there. They don't seem to eat it too much, but overall, and the product that they have, this everything I add seems to quickly become like a soil-like material afterwards. So it's very finished material, like it's like you could add it on the pot and grow some plants immediately. So this is box number one. I will show you box number two with my worms that I isolated from my indoor plants. Um, they are small worms, as I showed you before. Yellowtail looks like a Senia, but I don't think there is Senia based on the location of the clitellum. And I had a fruit fly problem on this box, a lot of fruit flies because it's very wet with the coffee and the watermelon that I added. So I'm slowly incorporating more and more cardboard into this box to try to dry it a little bit more. I haven't soaked the cardboard before because I want them to dry. So here's the worms, how they look like. Um, they have a behavior that if you grab them with the substrate, they seem to be very comfortable and extended, but if you grab it on your hands, they play dead immediately. Yeah, the substrate is full of also of cocoons, and they seem to reproduce very quickly. You can see a lot of baby worms in there if you look for. There are some pot worms in this box also, but they seem to be reproducing fine, and there's a high density at the moment. They increase the density quite a lot since I start gathering them. Yeah, so this is the worms from my indoors. There is some isopods in there also that seems to help on the composting process, the isopods. And I quite like the association of isopods and earthworms. I would try to set up with the student an experiment to test how much the isopods accelerate the work from these earthworms. So this is what I was talking to you about. If you put them on your hands, they play dead, they contract, they become like 2 or maximum 3 centimeters large. But if you water them, then they extend again. But if you keep them on the substrate, when you show them, they seem to relax and extended. So this is typical behavior when you see these worms. If you grab them with the substrate like I'm showing now, they will be extended and you can see their full size. But if you put them outside the substrate on your hand, they will contract and play dead. Yeah, they will contract and play dead. So this is the worms that I isolated. I'm also hoping that they will be useful for composting. They seem to thrive really well in these environments. I think coffee is, well, this is the rest of the watermelon. also decomposing quite quickly. So I will wait until the substrate dry a little bit more before I add more food for them. I'm just adding more cardboards as it goes, and try to control a little bit the humidity and the fruit fly problems. So next box I will show to you, or the box that are growing from the cocoons. These cocoons arrived exactly 15 days ago, and then you will see how much these worms are growing in 2 weeks. Last week I showed you some ecloding cocoons and baby worms, and you can see some bigger worms now as it goes. Bringing the box down. All right, so I added a little bit more substrate to this box because I seen the density was, it looked to me that the density was becoming too high, and I want them to have enough space to multiply here, to colonize and eat. Um, so so some of the baby worms you can see there. The bigger ones are 2 weeks old. Some of them are just 1 week. They're slowly increasing the population. Still, there are many cocoons that have not ecloded yet. And maybe they won't, but let's wait. So you can see here some of the worms. These worms are supposed to be Eisenia andrae. Also, I will confirm that when I do the identification process. The identification I will do both by visually, by with the microscope and counting the segments and looking at the male pore. Um, and also I will do it by sequencing of the cytochrome oxidase gene. So with that, it will be a more precise classification. And if I find there's something really new about these worms, I can even do full genome, and I will share with you guys the results from that. All with the objective of making scientific publications in our soil microbiology lab. So this is always in preparation for a course that I'm going to give next semester. The course is about waste management. So I want the students to work with worm composting. So that's why I'm doing this now and selecting these worms beforehand. The course starts in February, but during the next semester they will have to do something like I'm doing now. So before I give the students this task, I'm doing it myself just to make sure that everything is in place and they can do it, and they can describe their experience on this. And of course they'll have control pots with cold composting without the worms, and seeing comparing how the evolution of cold composting and the worm composting goes side by side. So this the last worms I have are the ones that are that I took from outside pot. They are also just ecloding now. In the fall, they might have 2 or 3 weeks or maybe 4 weeks old. They were completely absent on these pots during the summer, and now they seem to have ecloded when the temperature went down in fall. The summer here is over 45 degrees, so all the adults died, but the cocoons survived, and these worms all ecloded now. And I selected like 15 or maybe even 20 of them and put it on this pot, and I'm waiting them for mature a little bit more and get sexually mature, and I will try to classify them and know what they are also. They have a behavior different from the other ones that I have, that they're playing dead here, a little bit pale, this worm, but you will see its saliva will move in a bit. Yeah, there you go. They seem to be jumpy. Like like the red wigglers, the typical red wigglers. When you dig them out of a compost pile or a manure pile, they start jumping around and wiggling. These worms have the same behavior. So yeah, I'm hoping to see if they're good composters or not. Uh, Probably different strains because it's coming from the outside pot and not the indoors ones, and we don't mix the soils. Look at how they wiggle and jump around. This is what I was telling you about. There you go. So this is the curious behavior about this strain comparing to the other ones. And when I do the classification based on the position of the clitellum, and I will I'll make a video about it and show you guys how you can check for yourselves. If you have a good macro option on your camera, you can just take some photographs of that and try to identify these worms by yourselves just by counting the segments up to the clitellum and counting the segments up to the male pore, looking at the size and general aspect of these worms, and you can, based on that, you can arrive with a fairly accurate classification of these worms. So this is what I have to show to you today. Thank you very much, and see you in the next video.